Dendritic cells are a type of antigen-presenting cells of the immune system. They process antigen material and present it on their cell surface to T cells. Dendritic cells are found in tissue that has contact with the outside environment, such as the epidermis, linings of nose, lungs, stomach, and intestines. Dendritic cells are often seen patrolling the body, looking for pathogens such as bacteria and viruses. They engulf foreign pathogens and display its antigens, which are foreign protein fragments, on their cell surface membrane through MHC class I and II proteins. The dendritic cells then migrate to the lymph node, where they interact with B cells and T cells to trigger the adaptive immune response. Specifically, MHC class I proteins stimulates CD8 cytotoxic T cells, while MHC class II proteins stimulate CD4 T helper cells. A key component of dendritic cells that helps with the activation of T cells are co-stimulatory molecules. Dendritic cells maintain these molecules on their cell surface. These molecules help activate T cells and also act as a guidance for dendritic cells after antigen capture. Dendritic cells can also have some relation with B cells. Follicular dendritic cells help maintain memory of B cells. After the antibody response begins, follicular dendritic cells store antigens and antibodies. Follicular dendritic cells can then act as a storage of antigens for B cells, which take these antigens and present it to T cells for a faster immune response. Our bodies have shown to modify dendritic cells to activate an inflammatory or suppressive response. In the inflammatory phenotype, dendritic cells constantly monitor their microenvironment, taking up and processing foreign antigens. Once an antigen is captured, along with the appropriate activation signal, such as from pattern recognition response proteins, they mature and undergo several changes. This includes an increased expression of co-stimulatory molecules and the release of inflammatory cytokines. It is not always a good idea for the human body to continuously initiate an immune response. Dendritic cells can also suppress the immune system to avoid starting an inflammatory response to naturally produced antigens in our body. In the suppressive phenotype, dendritic cells will encounter antigens, which are naturally produced by the body, and present them as part of the maintenance of homeostasis. Normally, this results in the initiation of suppressive immune networks, causing T cell deletion, energy, and regulation. It can be seen that dendritic cells play a big role in dictating the fate of an immune response. For this very reason, it is often the focus of numerous biomedical experiments. Recently, scientists and engineers have been working on modulating the phenotypes of dendritic cells through biomaterials to either promote or suppress the immune system.
Biomaterials include any materials which are suitable for integration in the human body. These can include metals, ceramics, and natural and synthetic polymers. These materials have already found usage in regenerative medicine, providing structural support and facilitating host cell and tissue interaction. Active molecules can be incorporated into a biomaterial scaffold to deliver them to a specific area, and in many cases, biomaterials will be naturally degraded and absorbed into the body after their use. Among these materials, synthetic polymers have shown promise for facilitating modulation of dendritic cells to manipulate the immune system. There have been a wide range of synthetic polymers studied for their use as biomaterials. One example, a group of materials called polylactic coglycolic acid, or PLGA, which are copolymers of glycolic acid and lactic acid, have been studied the most for usage in immunotherapies. An inflammatory dendritic cell response is useful for vaccines. Vaccines, which can be either preventative or therapeutic, generally include three key elements: an antigen to trigger an immune response, an adjuvant, which is a substance that enhances the immune response to the antigen, and a delivery system, which is most often a liquid solution. Currently, the most commonly used adjuvants are a group of inorganic particulate materials referred to as alum, which serves both as an adjuvant and an antigen carrier. They are responsible for engaging the immune system and aggregating antigen. Through supplanting alum as an adjuvant or carrier, scientists hope to improve the performance of vaccines. Biomaterials can be used to form scaffolds, which allow for a long-term controlled release of cargo molecules, such as an antigen, by loading the molecules onto or conjugating them into the scaffold. The scaffold protects its cargo, which can also include other chemicals, such as activation signals, from degradation, and can be engineered to release them under certain conditions over extended periods of time. This can increase the likelihood that antigens are taken up by dendritic cells, and potentially eliminates the need for booster doses in vaccines. In addition, these polymers can be carriers for antigens that are too small for existing carriers, such as alum. In studies, therapeutic vaccines based on PLGA scaffolds were shown to be effective in treating cancerous tumor growth in mice through persistent release of a dendritic cell recruiting molecule, GM-CSF, and providing a structure in which dendritic cells can reside in while receiving the cargo of tumor antigens and immune activation signals. In addition, biomaterials can also be used in a particulate form to deliver antigens to dendritic cells, allowing for a higher success rate in the delivery of vaccine components. By attaching certain receptor proteins to the biomaterial microparticles and modifying the properties of the material used, certain internal components of dendritic cells could be targeted, leading to a better delivery of the cargo antigen to the MHC1 pathway. Finally, biomaterials can act as adjuvant carriers, delivering adjuvant molecules such as lipopolysaccharide and monophospholipid A, which cause upregulation of MHC2 and of co-stimulatory molecules, as well as the secretion of pro-inflammatory chemical signals. Biomaterials can even be engineered to directly act as adjuvants, directly stimulating dendritic cell activation, though this aspect of them is still relatively unexplored.
Dendritic cells can also be modulated to initiate the suppression of immune responses. As mentioned earlier, dendritic cells can support suppressive functions through T-cell energy in which lymphocytes become hyporesponsive, leading to weaker responses. This holds great promise as scientists can modify dendritic cells to a suppressive state, opening up possible solutions towards autoimmune disorders such as diabetes and arthritis. Many immunosuppressive drugs, such as dihydroxyvitamin D and rapamycin, can decrease surface expression of CD40 and MHC2 molecules in dendritic cells. Soluble administration of these agents have severe limitations, as it is hard to control dosage, and there is also no guarantee that these drugs will get to the targeted area. Specifically, when rapamycin is given in high dosages, it can cause systematic immunosuppression by targeting other lymphocytes. Use of biomaterials have opened up viability of these drugs as they can provide targeted delivery and controlled release. Using PLGA particles, doctors can safely deliver donor antigens combined with rapamycin to induce what is known as transplant tolerance. This will lead to reduced allergic hypersensitivity disorders through inhibition of T-cell activation.
Dendritic cells are key in identifying threats and guiding immune system responses. Biomaterials have shown great promise in studies at targeting dendritic cells for new immune system treatments, inflammatory and suppressive.
Thank you for watching.